first of all, good morning to you all, and it's great that we'll be having great conversation regarding the Seahorse Wave software. So this software is just not for setting up your SE, but this also acts the kind of a controller which connects with your system. And also post done, you can do all your analysis using the Seahorse Wave software. And first thing that it's a hard and fast rule that this software can also be installed on your own PC. So you can design your code template right on your system, like right on your PC, and then export it to the new software which is installed on the controller, which is right beside the Seahorse system. So, like I will step by step guide you how to even do that. But before that, we will have a general overview of the user interface of the whole Seahorse software. So, as you can see the screen, you will find that on the very left side, you could see the templates which are available, and just on it down you could see the result template. Once you guide to it, you will find all the results files which have been run on the system. Then the catalog part will guide you like how we'll be creating your code catalog for all the compounds, all the inhibitors which you will be using on your cell line. And the fourth option is your general directory of where your folders, where your files will be saved, and the help. So if you generally switch on your software and the Seahorse Wave software, at the here on the left side, you will also see that the system will try to communicate with the controller. Will try to connect it, try to connect with the Seahorse system, and it will display the software like the temperature of the system, which is like 37 degrees Celsius. Generally, for most of the applications, it will be shown on the bottom left side. So right now, what we are seeing is all the templates which are available for on the Seahorse system, like all the experiments which you can run on a Seahorse system. So you can see that the very first that's a blank template. So if you just want to generally see the OCAR and ICAR response for a particular drug, you can do this. You generate it using a blank template, not running any specific kit like microstress kit.
Audio problem is there. Sometimes it's breaking. Hello.
Hello.
The audio issue is there. Sometimes breaking audio.
Is it good now? Yes. Just okay. I hope it's clear now. Hello.
Yes, fine.
তুমি এখান থেকে তাহলে অ্যাটেন্ড করবে, আমি তাহলে অফিসের কাজগুলো করি, ঠিক আছে? এটা আনমিউট করবে, আমি চেক করবো it is running or not, because this will only have.
In the Seahorse portfolio itself, there are many number of kits available to run specific assay. Like if you could recollect the last presentation which we had on Seahorse system, so there were many assays like right from the beginning you can do the ATP rate assay, which will tell you if your cells are more dependent on mitochondrial respiration, on glycolysis. And as you go down there, you have multiple assays like microstress assay, the glycolysis stress assay, the glycolysis rate assay. And other than that, there is also a study the dependency and flexibility of your cells on a particular substrate like glucose or glutamate or pyruvate as such. So the templates for all these experiments are readily available on the Seahorse Wave software. It's just you have to select one and start running your experiment. So right now, I will be showing you how to design a template using the XF cell microstress. You just have to click and open this particular template. Okay. So at the very topmost part, you will see the very first thing which we have to define are the group definitions. The very first thing. The group definitions will be actually deciding what will be our injection strategy, like which compound will go into our group A, port B and port C. Next, since we have already decided that we will be running self microstat acetate, this injection strategy, one single injection strategy will already be done. Now, if I click on this, you will see here on the injection conditions that on the port A there will be oligomycin, which will have a specific concentration. Similarly, in the B port there will be FCC. Now, concentration here is not defined because FCCP, while you are running the microstat acetate, needs to be standardized or needs to be optimized as per your cells whichever you are using. Because not one single concentration will work for different cell lines. So you have to optimize it on your own. And that can be done in a single experiment itself. And that's why you can design multiple injection strategies in a single experiment. I will show you how to do it. And similarly for rotenone antimycin, it has a fixed concentration. You don't have to do any kind of optimization here. 0.50 micromolar concentration is suitable for all different conditions or all different samples. So, when we are going for group definitions, first what we have to design is our injection strategies.
I have a question actually. If you
Yes,
go
yes.
to the C section, C.
Yes,
C. So the rotenone and antimycin A, is it 0.5
Yes.
micromolar when both together or each one 0.5?
it's actually in the microstat state, it's a combination. It's a mixed powder.
So this is
final
basically
concentration
peak.
goes
So,
for yeah,
each
this is a
one
combination.
concentration not different.
No. This is just one single vial with the rotenone and antimycin mixture.
Okay.
That's why. So it's like a final concentration for both combined. Okay. So now, if I would go here, my first compound is oligomycin, which is 1 micromolar. My second compound is FCC. Just say here also I will be adding FCC 1 micromolar. Okay. Now, rotenone antimycin, the concentration will be fixed 0.50 micromolar. What I will do here is I will just rename this condition as 1 micromolar FCC. Now, here on the injection strategy, you will see a tab for add, which means another injection strategy will be added. Now, you just have to go here and just rename this injection strategy to 2 micromolar. So what I will doing here is now in port A, port B and port C, since we have added another injection strategy, it is not defined. It is not readily available like this. So what we have to do, select port A, add compound. Now you have to go here, select compound catalog, scroll down and you will see here there is oligomycin already present. Now select oligomycin. Select concentration of your interest. Okay. Now this our port B is done. Now we'll go to port B. Select add compound and then I will select FCC here. Now since I am increasing the concentration of FCCP for my another set of group, because I have to fix concentration of FCCP once per percent. This gives me the maximum respiration rate for cells. So I will make it here 2 micromolar. Now one thing here you can use is all the dilutions of oligomycin FCCP is done using Seahorse acid medium itself, which could be either EMAM or RPM. So those details can be added. This is optional, but if you want to follow a good practice, you can write it down here as Seahorse except EMAM. Now this can be done for all the compounds. Now port C, again I will go add compound from the catalog. I will select protein antibodies. Now the concentration here is 0.5 micron. Okay. So now you can see that I have designed two different injection strategies. Okay. Now here the concentration on oligomycin was fixed. The FCCP concentration was varied. Similarly, if you say that I have to design another injection strategy wherein the concentration of oligomycin will be different. Now, since here it was oligomycin 1 micromolar, okay, I will make it something like 2 micromolar oligomycin. They indicate here right first just for your own understanding, so you shouldn't forget which injection strategy defines what. Now, if I say 1 micromolar, I know that here FCCP is 1. Here, like which the condition has been different than the other condition. So I know this here oligomycin is 2 micromolar rather than 1 micromolar. So I will go here, select compound oligomycin, I will make it 2 micromolar. Similarly, here I will go select an FCCP, indicate 1, and we add compound. Protein on antimycin, it's 0.5 micromolar. I will add another injection strategy. So there is no limitation as such how this injection strategy we could implement. So again, compound catalog, let's select oligomycin, we'll take it to micromolar. Add compound FCCP. Here I will make it to micromolar of FCCP and protein on antimycin 0.5. Now we will go one by one. So our first strategy has 1 micromolar of oligomycin, 1 micromolar of FCCP, and 0.5 micromolar protein. This was one condition. Now, second condition, the same concentration of oligomycin, but a different concentration of FCCP, and the same concentration. Here, again, we had a one concentration of oligomycin against 1 micromolar of FCCP and protein. And here again, oligomycin was 2 micromolar. Here also it was 2 micromolar, but we had combined it with 2 micromolar of FCC. So that way we will be able to test our 4 different conditions depending on the concentration of oligomycin and FCC. Any doubt here?
No,
Hello?
it's fine.
So once your injection strategies are defined, there is a second option called pre-treatment. Now pre-treatments is the option when you treated your cells with any kind of drug or inhibitor 24 hours or maybe overnight before your experiment. Now just say yesterday I seed my cells, there was a control group and there was experimental, and today in the afternoon I will be treating my cells with just say an anti-cancer drug like doxorubicin, and my cell lines will be something like MCF cells. So I will here create a control and experimental group. Control will be treating with something like there will be no treatment as such, but with experimental you could like put down inform like create a description like it talks about this. Like just a short description here, and so this way those your control and experimental will be different. Now in essay medium, you could select which media you are going to use in your essay. For say you are using Seahorse, it's a DMEM medium. So here you could see the compositions. Like these are the standard composition of a DMEM essay medium which will be used for your Seahorse essay, which include 10 millimolar of your glucose, 2 millimolar of your glutamine, and 12 millimolar of your pyruvate. Now pH will be 7.4 for all the essays. You can also mention in date as in when it was prepared, 7th, and prepared by, just say, if I would just write down an quick spread. So this is. So our injection strategy is done, pre-treatments is done. If you don't have any kind of pre-treatments available in your cells, or you are just simply running an assay without any kind of drug treatment, then you can just delete these things. Okay. Just you have to make sure that on the right side you have to also delete it from the groups. There should be only blanks available. Now I will go to my cell type. Here we will be adding details as such, which cell line we are using, what is the density of our cell line, and prepared by whom, what is the passage number which we are using. Source and lot number is optional. If you have those details available with you and if you feel that those should be added while you are setting up your assay, you can do it. Now, if you can see, some of the basic cell lines are already available in the control. So just if I scroll around, you see that I already have FG2 and MCF cell. So cell type, I could just write like adherent. What seeding density I will be using? Just say 9,000 or maybe 20,000. So just on a safer side, I'll be also writing mentioning here like 20k. Now lot number, if I have those details prepared by friend, just passage number 6, prepared on just say as thought it like last week. So yeah. Prepared on just gave them a last passage last week. Similarly, the similar setup I'm adding another cell line, you can say. See E549. Here also the same 20k. Cell type again. Adherent. Seeding density 20k. And prepared and all this. So this way you can again add as a
excuse me. Hello.
Yes.
Yeah, I have a question actually. So here, what we are doing is that one, we are just taking adherent cell lines. So we are using the DMEM media, and we have like written that we have taken DMEM media, right? When we were doing the dilution and like solvent percentage and everything.
Yes.
So, for example, if I want to work on, if I'm doing a standardization first and or any sort of experiment I'm doing where I'm taking like both adherent cells and suspension cells. Okay.
Suspension
so
cells. Okay.
yeah, for example, if we are doing like MCR7 and we are doing K562, That's a leukemic cell line, right? Suspension cell line.
Yeah.
Yeah. So here I in the MCF7 cells, I'm going to give my DMEM media, and in the suspension cells, maybe I'm going to give RTMI media. Yeah. So the thing is that here, the assay media and the injection strategies where we are giving the media, we are setting these for all the cell types, right? So how will I be able to differentiate the media name out there? And if I'm not being able to, is it going to make any difference?
Yes. Here really you can't use a single, two different medias, because in our blank wells you have to one specific media, because the system also calculates the buffer factor, which is really important when we are going for calculating the extracellular acidification of the cells. And our baseline has somewhere has to be maintained on the single cell, where we will be adding only our media. So just adding two different medias and which are having two different compositions, it will be like kind of messing up with the baseline of our results, and that is generally not recommended.
Okay, thank you so much.
Yeah. So generally when we are going to set up an essay, I would just say that if for 1 or 2 hours of experiment using Seahorse media, will it be making any difference in your cell line? You could see that if you are doing your cell lines on RPM, but running your essay on DMEM, but it is generally not recommended to use two different medias. Two different cell lines, yes, but again, if they are like grown in a single type of media like DMEM, then yes, you can do that. But if those cell lines require two different medias, then it's not right.
Yes, yes, thank you so much. Yeah.
Because we have got this kind of questions and we just have tried to implement that. But the background actually which we got and the variance, we could really understand that the things on the background are really not doing good when you are using two different type of media. Okay, so now once all things are done, like we have defined your injection strategies, we are done collect pre-treatments, it's optional, or if you are having a controller experimental book, you can keep that. We have defined our assay media, and we have also defined our cell type or cell lines which we are going to use on our experiments. Now, I have to just do a simple thing. Here, you will see an option of generate groups. I will just click on that. And you can see on the right side, there are different groups created. So there are 1, 2, 3, 4, 5, 6, 7, 8 different groups. So you see, one injection strategy for one cell line, like all these 4 injection strategies have been implemented to MCF720K, and like all this injection strategy have been defined for T540. So that way, you have got 8 different groups available here. Everybody get this, like how those groups are created? Hello, am I audible to you all?
Yes, sir.
Yes, sir.
So now, once I am done with my group definitions, I will go for treatment. Now first thing first, on the right side you can see there are lines already been defined, like A1, B4, C3 and B6. These are predefined lines. Yes, you can do change them, but in many of the cases it is not recommended. But you can do keep them on one side. Right now, I will just keep them as it is, and I will start to design my treatment using all 8 groups which are available. Now just say there are 8 groups and we have 20 wells available. So for just say for the microstress 1 micromolar FCC 20K, I will kind of add 3 groups. For this particular group. Again, I will add it. You can also drag and drop. So it's calculation on my end. So for the last group, I will just keep it 4 minutes. But it isn't supposed to happen on all your end. You have to keep equal number of replicates in your experiment. So this is how you will, once you have generated your groups, you will define your plate maps. Okay. Now, once you are done with group definitions, then plate map, then we will go for our protocol. Now, this is a set protocol for the templates which are already available, and this is kind of a standardized. Like every experiment, the very first thing, equilibration and calibrates are really compulsory and recommended. So those will take their own 20 to 30 minutes before the start of the essay. And after that, you'll have a duration like 24-minute cycle of your baseline readings. These baseline readings are taken before injection of any kind of compound. Now these 24 minutes are divided into 3 different cycles. Now each cycle will have 3 minutes of mixing, 2 minutes of wait time, and 3 minutes of measurement. Mixing because whereas you can also make changes in the protocols. Like since we haven't added any kind of compound, just say you can decrease the mixing cycle. Like 1 minute of mixing is enough because there's no compound added, and even wait time can be brought down, and measurement is 3 cycles. Now you can see as I go on to increase and decrease the timings for every stage, it affects the total time the experiment will be running. Like I say, if I make it again standard, like 3 minutes, it goes to 1 hour 45 minutes. You can also increase the number of cycles. Like instead of 3, we'll go for 4 or 5 measurements per cycle. Again, the total time will increase. Similarly, after your 24 minutes or 21 minutes are done, your first oligomycin compound will be injected. And here, again, the cycles are fixed. Here, it's better to go for 3 minutes of mixing, or maybe you could make it 2 minutes. The standard is 3 minutes. And 2 minutes of wait time, just for things to cycle, and then 3 minutes of continuous mixing. Even here, you can increase the number of cycles. And the same goes for your accessibility, and the same goes for your product. Okay. Now once this thing is done, we will go for your run essay, wherein you will be writing the name of your project team, who has created it, the name of the scientist, project. These are all the details related. And if you have any kind of note, and here you can say it's a bond.
Hello, I have a question.
Yes. Yes.
Can you go back to protocol section?
Yeah, protocol.
That measurement cycle, 3 minutes is there. So this machine will measure 3 minutes continuously.
Yes, this machine will measure 3 minutes. But
That's my question actually.
In what sense? Yes, and it will plot an average of that. Like on the screen.
Okay.
Once I will share even we will even look at one data. So the measurement in one single cycle it will go on for 3 minutes, and an average will be plotted along with the error bars.
So average over 3 minutes data.
Yeah. So one cycle, 3 minutes of continuous measurements.
Okay. Fine.
And I have a question that
Yes.
if we are using our compound, like there is a blank injection which is deep
D4. So in D4,
Yes, yes, yes.
if we are using our compound, so for that the parameters will be by default that 3 minute, 1 minute, 3 minute.
Yeah,
Because we
I
don't
will.
know what is the. So
Yeah.
this,
Like for that,
this will be this will be better to go with the default system for standardization, right?
I will give you because there is another template which is already available for any kind of compound you are using, a compound of your interest you want to introduce into this mess. So now this is once it's done, you can run the essay. Then here you will get an option for since my own software is not connected to the system, so you will get an option for start essay. So once you click that, it will ask for your calibrate rate. Once the calibration for 20 minutes is done, then it will ask for a culture rate. Meanwhile, you also have to add your compounds or oligomycin FCCP into the ports which are available in the cartridge and start with. Now, as Sir said that he has a compound of his own interest, so you can see the name is except cell mitostress test. On the very right side, you will see cell mitostress test acute injection. Now, acute injection is a protocol which involves a control and experiment at the very beginning. Like if you see you are using any kind of inhibitor, so you do that maybe 5 or 10 minutes, it is or maybe instant tendency. It will block mitochondrial respiration or say the pathway. If you have any such kind of compound available with you, so here you can see there are injection strategy already available. Like first thing is control plus mitostasis. If I just click here, now in the port A here we don't have oligomycin. Instead there will be a control compound. Control compound could be anything. Like maybe if you are using any kind of vehicle control, then you could add EMS loop or maybe media itself. Like just say I'm just going to add media. Like the Seahorse except media. Now in our port B, we will be having our oligomycin. So this is where the mitostress test gate comes into picture. So here I say first your compound of your interest will be added, and then the compounds which are available in the kit itself. So the pre-port we have oligomycin, FCCP, and rotin. Similarly, if I go to experimental compound, so there are not much drugs available like say paclitaxel. We will add a drug like paclitaxel, which I know it doesn't act instantaneously, but I will just add this detail in what compound, what concentration I will use. So paclitaxel will be my compound, then I will have an oligomycin, an FCCP, and then my rotin. Sir, does this answer your question, like what you were exactly looking for?
Yeah. Thank you.
Yeah. So for always remember that when you are going for like introducing any compound of your interest, go for acute injection, wherein you can readily add your compound of your interest in the first one. Now everything here remains the same. There is no difference. You will
Sir,
have
I have a question.
Yes.
In the previous injection strategies, you were mentioning for pre-treatments, control and experimental
Yeah.
groups. So if I add control as well as experimental groups, then how these groups would be divided?
Now, so there will be multiple divided. So here is what we do. Since we are here optimizing the concentration of FCCP and polymers, so in this particular set of experiment, won't be using any kind of we will just simply add our cells, maybe in a number of concentration like 10,000, 20,000, just to see what works for us and what concentration of oligomycin FCCP works for us. Once I know that yes, 5 micromolar of oligomycin works for me and 2 micromolar of FCCP works for me, then I will go to I introduce my control and experimental group to the next set of experiment.
Okay.
Got it. So right now, just for the sake of explanation that how different kind of infections are that's why I just created different concentration of polymers. But once I know that specific concentration of those compounds work for me, Then I will, in the next set of experiment, I will have my compound like add a pre-treatment of vessels. But while we are setting up the concentration of these compounds, the optimization, it's better to do those set on those kind of optimization without treating cells with any compound, like any drug.
Okay.
So this will be something kind of my preliminary experiment when for the very first time I will cell line. So example maybe A549 or MCF7. If in literature I would get all those details like which oligomycin FCCP works, then you can directly go for your experimental kind of drug related experiments. But if we have no such data available with you, then you have to optimize it first and then go for drug. So there will be always first optimization experiment and then drug treatment. And if your compound acts instantaneously, then you can go for acute injection. Where in the port A itself, you will add your compound of interest. Now, if I just go here and catalog, you could see here there is an option of add compound. Okay, so here I will add such details. Like for just say there's a drug 5-fluorouracil 5, and what concentration finally I will get. Let's say for 5 micromolar or something. And what solvent I have dissolved it in. DMSO and what percent. Once you click, 5FU will be added in the catalog. If I just go to templates and this experimental drug treatment compound catalog, you will see 5FU is already available. I just have to go add 5FU. Just go back catalog. 5FU. Make the changes. Go back to template. Select from compound catalog, template 5. So this way you can add all your compounds here in itself. Like any drug which you are going to use today or even in future experiment, you don't have to kind of add it separately. You can keep them in the template itself. Similarly, in pre-treatments, medias are already available. You can add your even cell line of your interest. You can keep it in the catalog. So this way, if someone is going to use that particular cell line in their future experiment, they would find that particular cell line in the catalog itself. So no one has to add it separately. So this kind of works as a library for all. Okay. So this is how you design even your catalog for
So,
compounds.
One question that if we are using new media type, so for that case, what optimizations do we have to perform for that particular media?
So mostly most of the cell lines, like for Seahorse experiment, we generally recommend Seahorse experiment. There are certain categories. This was like
Sir, your voice is
even in the experiment.
hello. Sir, your voice is not clear actually.
So, Pranav, actually, I am also having difficulty.
Yeah.
Sometimes the voice is getting low and not able to understand.
Hello.
Hello, can you listen? me?
Can you hear me now?
Yeah, so your voice is kind of breaking actually, and also a lot of time I'm not able to understand.
the network is down in his.
Now it's clear? Hello.
Better.
Now it's
Better.
Can you hear me? Hello. Hello.
Yeah, it's better.
Yes, we can hear you.
He may be having some difficulty in connection. Actually, somebody is recording this whole thing, right? Somebody is recording it.
Yes, sir, it is being recorded.
Okay.
So,
Can you hear me now? Hello.
Yes, we can hear.
Yes, yes.
Yes, sure. Well, let me share this. Yeah, so like I was addressing the question for different media and optimization. So, like I generally recommended to use the Seahorse XL medium itself, but if you face any kind of problem, like if your cell line, or per se if you are using isolated mitochondria, which requires a specific medium, I guess MEM medium or something, then you have to optimize it yourself. And first thing you have to make sure is the pH of the medium should be pH 7.5. It should be no more than that and no less than that. Composition, you can take it on your own. There are protocols available. Maybe even I could share it with you. But the most important parameter here is your pH. It should be 7.5. Because if it's any kind of mix up in that, then the extracellular acidification data which you are getting won't be that appropriate.
Okay, so only thing is we have to maintain that pH, and we have to give a blank when we
Yeah,
we
that
run
should be our also that's specific. Yeah. And just make sure that pH, that particular media doesn't have phenol red, or it might interfere with the release.
Okay.
Sir, I have a question.
Yes.
So, the day before the experiment, we have to actually set up the instrument so that the temperature reaches a particular temperature. So the instrument reaches a particular temperature. So for that, what should we do? First switch on the instrument and then will any settings come up?
There's no setting. You just have to turn on your system, and the wave software which is available on your controller, you also have to switch it on. And before going, just like if it is displaying the right temperature, 37 degrees Celsius, and let it equilibrate overnight. And if you are going to do your reset tomorrow, just before going like late evening, just start the system and let it equilibrate. So you don't have to do anything. You don't have to clear all the settings. That's it. Just use the main power button of the system. The system will take care of it, and thus open the software. Keep the software open over it. Nothing else.
Okay, sir. And the temperature 37 degrees Celsius is required for only cell lines, or can it be for isolated mitochondrial bacterial cells?
Isolated mitochondria, I guess, requires a lower temperature of something, maybe 20 something, and you can change the temperature. The option is available.
Okay. So for that the room temperature will also matter, right?
Yeah, the room temperature will matter.
Okay.
So it should be like if you are setting an experiment, it should be 10 degree lower than what we are using the system for. Like per se, if we are already having 37 degrees Celsius, then the room temperature should be around 20, 22. That. So you
Okay.
can just template it sitting here.
And before starting the actual experiment, so we have to calibrate the plate and
Calibrate the
then
plate. Yes.
equilibrate. That will take like 20 to 30 minutes.
Yeah, 20 to 30 minutes max.
Okay.
Hello. So I want to question with last time we had this meeting, so we suggested that we would share the protocol for coating the plates with PDL for non-adherence. So
Suspensions. Okay, I will share it with you. I do have a protocol with PD. You can do it with Santac, PDL, or EL. So I can share with you. Like I do have a little bit protocol. I will share it with you after the session. So this was the part of your designing your template. So just say we can save this template particular. Yes. Let's see. Let's see. Now once I save it, if I'm going to go, that particular template is available. Now what I can do is I will select it and I can export it and save it on my phone. Now I have exported it into my laptop. Just see if I would go here, import documents, and this particular template is available here. I will just select. So it's already available, so just say just remove this, import documents, ISO and I will import. So you can design your template on your own PC, take it in your pen drive, just connect it to the controller and directly export. So all this designing template, you don't have to sit on the controller itself and you can do it on your own desk. So that's not a problem. So once we have done with the templates, you can go for your results. Just open one single file here. So this is how your data for microstress SE will look. So if you go here, there is a quick view. So this is your quick view. The OCAR and ECAR data will be parallelly shown. So this summary. This is just kind of a printout where you go add all those details. But this is not generally used. Data point of view, you will get all raw data, like what was the rate of OCAR, ECAR, the level of O2 and pH, the raw data. This is something just to, for us, to check if the system is working fine or not, but it will just show you the what was the bed temperature, and the parameters which are showing here is just when the system was calibrating, just to see if the O2 and pH sensors are working fine or not. Same goes for your calibration. The data which is important for you will be here. It has been arranged. So, that's also overview. So overview wise, you could see there's OCAR is represented. If you want to display it now, here 3 different groups are available. Now if I have to arrange it well wise, even I can do. And if you want to change the axis, that is the process. Like instead of rate, I have to check the level of oxygen, how it has been moved now. but majority of the time your experiment will be dependent on group wise. Here is your OCAR data. Now if I just make it ECAR, so these are my. Now as you know that you have to also normalize your data, the option is available wherein if you are using doing cell count, and you can just directly put in like let's say I have added 20,000 cells. So this is my bank actually. So there will be no count here. The same I have added 10,000. Just say like 19,000. This way you could just add the values and click apply. So your normalization will be up. Similarly, if you are going for protein estimation, you could just like put microgram and however much of protein you have got, just for example 20 kg, 18 kg, total well protein. You can put those values here and just click apply. And your normalization will be done. Here there is an option of export. If I would just select that, you will see the raw data which was available here, you could access it on an Excel sheet using this particular format. Just say if I would Excel it to transfer it to Excel file, just replace it. It has been done. Go here. So, column wise, group wise. And you can also export the graphite prism file. What you will get in graphite prism is first I will show you the report generator. Now here you can see there is a report generator for real-time integrated test, algorithm, test, and cell microstress. This is of our importance. The microstress report generator. I will just click on it. And I will simply because I do already have this file, so just that I will search. And I will show you how the report works. Sometime. So here it will ask you what all groups are available. So I will just select all 4 groups and I will just click update submit. So you will find all these graphs available. The basal versus respiratory and bar charts wise you will get your basal respiration difference between all the different groups. How much of proton leak is contributing to your basal respiration, what is the maximum respiration capacity, difference between those groups, spare respiratory, the non-mitochondrial oxygen consumption, the ATP rate production rate of your cells, coupling efficiency between ATP rate by mitochondrial respiration and glycolysis, and how much is the spare respiratory capacity. Now all these graphs will be also available on your graphite disk. So whatever data which was available here now has been converted into graphs code. So even if the raw data is available, you don't have to do any kind of calculation as such, no graph plotting. And if you have to understand how those details have been calculated, so you can find it here in the sample calculations, like for mitochondrial, non-mitochondrial oxygen consumption, or for basal respiration, what all measurements were used. So it was like the last rate measurement before first injection minus non-mitochondrial respiration. is also shown here. Like, this is just a representative data, not from the actual experiment. But if we just go here, every measurements for every group, it defines this. Like first three measurements I know, those are for my baseline. Next are my oligomycin. Next FCCP and protein. So from here you can pick one value, do the calculation, and you will get all this available across. So the only tough part is the setting up the experiment, and after that the system will take care of all your data. So option for that you will get. Similarly, there are also many other report generators available. If you are anytime going to do the experiment and those are not available on the Seahorse controller, which has been installed in ISA Kolkata, I could share the file. You just have to import it and share the steps. But these are the basic report generators which are already. So you can, most important here is the report generator and even the graph. Any question?
Yeah, actually, I have two set of cells. So one is control, one is already treated. So if I want to check the kinetic measurements, I don't want any injection ports or anything from the kit. So can I go for that blank sheet?
Okay. Okay.
Hello.
Yes, you can go for the blood experiment. Yes, you can go for the blood experiment.
Can you please
Yeah.
show me once the blank experiment?
Yeah, yeah. You just don't have to define SH. You can just hello.
Hello, I'm not
Can you hear me?
getting it
Hello.
clearly. Hello.
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 Yeah. So I have already opened one.
Huh.
So no injection setting. Which is first there is treatments available. Yeah, hello.
Hello.
Can you see my screen? Hello.
Yeah, I can see your screen.
Hello. Hello.
Hello.
Hello. Can you hear my hello? Hello.
Yeah, I can hear it.
So I'm sharing.
Network is there.
Sharing my screen for that particular thing only.
No, you
How
are
you can set
already
up your
that your
The screen is not
yeah,
on.
now
experiment
your
blank.
screen is visibility. What?
Hello. Sir, your screen is stuck.
Hello.
Yeah, your screen is stuck actually.
Yes. So
we can't see anything. Hello. Please you can reconnect if possible.
hello.
Hello.
Hello. Yeah. You can see that here I already defined control and experimental conditions. I have defined one single cell line and I have got my two different groups, control and experiment. Okay. Then I went for my plate map. I have just added the groups. And protocol, I have only kept the baseline. Only baseline just to see. And I have only increased the number of cycles. You can also keep that low, but I feel like since you are only doing baseline with no like injecting any kind of compound, you can keep it like 6 cycles and just see over 1 hour. Just in 1 hour, what will be your baseline between different groups. So that we can also include your like untreated cells, the vehicle control, your experimental condition like drug. If you are including 2 different drugs, you can keep adding. So that way 4 or 5 groups will be created. And just add it on the plate map and directly go for an experiment. So that's not a problem. You can that other things like getting production and all, calculation will not be possible.
Yeah.
Give you the idea about your pixel or Ukar and Ikar.
Yes. So I can just take the data and manually calculate it, right?
So here is basically use the kit for calculating all those parameters. So the black experiment is not.
So I will get the kinetic measurements.
Your kinetic measurements you will get.
Yeah, that's what I'm saying.
Yeah, that's that raw data you will see.
Okay.
Can't do it part from the normalization part. You have shown just to me back. The normalization.
Yes.
Hello. Can't do it part from the normalization things like we have shown previously. The protein normalization stuff.
For the black experiment?
Yes.
Yes,
Yeah.
you can. Yes, for that you can.
There is an option in normalization. I think I saw it.
Yes, there is an option.
Please can you show it again for this experiment?
So here, put here the top normalize.
Like after you will get the data, then you can do it.
Yes, then you can normalize.
Ah.
Not in there. Now I need to I will addresses measuring. Is there anything else?
One thing that Do you have any template regarding this data processing and protocol setup? If you have, I think if you have, you can share it with us.
Yes, sir, I do have
And
okay, I do have a manual.
yeah, so you can share it. And another thing, so basically this system is used for mammalian cells, but nowadays where people are using for bacterial cells as well. So if you have any template regarding that protocol, so if you have like this kind of templates for bacterial cells. Hello.
With bacterial culture work, there is like one single inhibitor doesn't works for different bacteria.
I
If you are-
cannot hear you clearly.
Hello, hello, can you hear me now? Hello.
Yeah.
Hello. So with a bacterial inhibitor, I could do share a protocol, but the thing is, therein you require a different media for your bacterial
Yeah.
culture, and again, the inhibitors will range a lot. Like a one single inhibitor work doesn't work for different bacteria. So we have
Yeah, that is fine.
Yeah.
But that is fine, and that's why I have already asked about.
I have I do have a
the
raw
media.
protocol for that. I do have a raw protocol for that. I could do share with it. So like how to set
So
up.
please, if you have, then please share it.
Yes, yes, yes. Sure, sure.
Thank you.
Thank you.
Sir, I have like we are also doing in bacterial cells, so I have prepared a short presentation regarding how to adhere it and all that stuff from the papers available. So can I send it to you?
Yes, sir. Do share.
Okay. Okay. Fine.
I will give it a look and
Like, can you just give us some guidelines how to optimize it a little?
yes, sure, sure,
Thank you. Thank you so much.
sure. We can do that. Hello.
Yes.
Hello.
Yeah.
Hello. Yes. Yes. So I wanted to know, like, will you be able to share like a very generalized protocol of maybe the cell seeding numbers for like adherent cells and suspension cells? Will that be possible? And the protocol for the cell adhesion for the suspension cells, like shown recently.
Okay.
Yeah.
So, like suspension cells, you want a protocol for that?
Yes.
Okay.
Yes. Yes. Yes.
Okay. Suspension cells is already available with me. I could modify it for you and I will share like that.
Yes. If you could please. Yeah. Thank you.
Hello. Yes, Tommy. Hello.
Yeah, we can hear you, but I think Somita is not there. So
That's why, since we are address all the questions, maybe we will end this call for today. And if you have any kind of doubt, just drop a mail anytime. And the questions which we had regarding protocols and all, I'll be sharing it with you by today. Of course, for bacterial part, I will need 1 or 2 days, so I could share it with you by end of this week. But Ethernet and suspension cells, there is a protocol available, so I will just share it.
Okay, please share it, and I think we are done for this session. And
So thank you so much for attending this session, and it was really great to interact with you all. And I hope this session helped you, and you will be much more confident when you will be using the Seahorse system and the way software.
Okay, thank you so much. And we would be knocking you again if there is any problem.
No issue with that. No issue. You can contact anytime. You can drop a mail, and if you are like kind of optimizing any experiment, just keep me in the loop, and I will.
Yeah.